It's a pleasure to be with you. The title of this talk is Environmental Public Health Practice, Part 3, Toxicology. There are no disclosures for this presentation. Objectives include review the United States toxicology laws, define the discipline of toxicology, review the history of toxicology, discuss the dose response concept, review the 5 toxicity factors, review toxicity testing methods and the 5 steps to approve new drugs in the United States, review toxicity risk assessments and perceptions. As of 2024, the Toxic Substances Control Act, TSCA, inventory of the Environmental Protection Agency, EPA, listed 86,741 existing chemical substances that are manufactured, processed, or imported in the United States. More than 60,000 of these commercial chemicals have been allowed on the U.S. market without safety testing. Even with significant safety data on a substance, the EPA must prove that it poses an unreasonable risk before it can impose a restriction. To prove that a substance is an unreasonable risk is a huge burden. It's difficult to define a threshold for unreasonable risk. For example, the EPA can only limit the use of versus ban asbestos, a known carcinogen demonstrated by numerous studies. Asbestos still kills approximately 15,000 people each year. The Frank R. Lautenberg Chemical Safety for the 21st Century Act, passed in 2016, requires that the EPA enhance the review of commercial chemicals for safety. This law was the first overhaul in 40 years of the Toxic Substances Control Act of 1976. At the current pace, it will take centuries for the EPA to complete its review of current chemicals, not including the hundreds to thousands of new chemicals introduced each year. A more robust and efficient system to review the toxicology of commercial chemicals is needed in the United States. Toxicology is defined as the study of the adverse effects of chemical substances on living organisms. The field of toxicology overlaps with the disciplines of physiology, pharmacology, pathology, epidemiology, chemistry, and statistics. Toxicology and epidemiology work closely together. Epidemiology studies the occurrence and etiology of health impacts, including substance exposures. And toxicology, the dose-response relationships and mechanisms of action of those substances. Toxicology assesses exposure, risk, and hazard. A basic assumption of toxicology is that all substances are poisons. The right dose differentiates a poison from a remedy. Throughout human history, humans have been fascinated by poisons. In Greece and Rome, poisons were used for suicide, executions, and assassinations. Socrates, 470 to 399 BCE, was executed by drinking hemlock poison. In Rome, Nero apparently dispatched Claudius with arsenic. During the Middle Ages, poisonings were a relatively common way to deal with difficult spouses and rival politicians. Paracelsus, 1493 to 1541, was a Swiss physician and is called the father of toxicology. He studied the dose-response relationship, stating that the effects of a poison are related to the dose, and that poisons target specific organs. In the 17th century, physicians became aware of the toxicity of industrial metals and chemicals. Matthew Orfila also influenced the field of toxicology through his book, Traits to Poisons, published in 1813, that described various poisons and their bodily effects in detail. The field of toxicology has several specialization tracks, including regulatory, forensic, clinical, environmental, reproductive, and developmental toxicology. Environmental toxicology is a multidisciplinary field of science concerned with the study of the harmful effects of various chemical, biological, and physical agents on living organisms. Ecotoxicology is a subdiscipline of environmental toxicology concerned with studying the harmful effects of toxicants at the population and ecosystem levels. Let's now consider a few key terms used in toxicology, including poison, toxic agent, toxicity, toxic substance, toxicant, and toxin. A poison is defined as any agent capable of producing a deleterious response in a biological system. Virtually every known chemical has the potential to produce injury or death if it is present in a sufficient amount. For example, common table salt can cause injury or death dependent on the dose administered. A study reported in the journal Nutrient in 2017 by Campbell and Train reported the fatal dose of sodium chloride in 35 individuals, 19 adults and 16 children. The lethal dose was estimated to be less than 10 grams of sodium or less than 5 teaspoons of salt in 2 children that died, and less than 25 grams of sodium or 4 tablespoons of salt in 4 adults that died. A toxic agent is any factor that can be harmful to a biologic system and would include heat and cold, ionizing or non-ionizing radiation, venoms, and almost all chemicals, etc. Toxicity is defined as the degree to which something is poisonous. Toxicity is related to a material's properties. For example, water, ethyl alcohol, and salt have low innate toxicity, requiring large doses to reach toxic levels, where dioxin, black widow's spider venom, and botulism toxin have high innate toxicity, requiring low doses for an adverse effect. Toxic substance is defined as a material that has toxic properties. A toxic substance may be defined as a single chemical like arsenic or lead, or may be a mixture of toxic chemicals like gasoline. Toxic substances may also be classified as organic or inorganic. Organic toxic substances come from living organisms and are usually large, complex molecules, where inorganic substances do not come from living organisms and are usually small molecules. A toxicant is a toxic substance that is man-made or stems from human activity. The toxic effect of toxicants may be direct or indirect. Potassium cyanide is directly toxic to the body, where methanol is only toxic to a human after being converted to formaldehyde in the liver. A toxin commonly refers to a toxic substance made by a living organism, including venoms made by reptiles and insects. Toxins are also made by microorganisms, like botulism toxin from Clostridia botulinum that causes foodborne botulism from inappropriately canned foods. Plant toxins include those from Amanita phalloides, or the death cap mushroom, poison hemlock, digoxin, a cardiac toxin from the foxglove plant, poison oak and poison ivy, and oxalates from rhubarb leaves, and the houseplant Dieffenbachia. Symptoms of oxalate poisoning include sore throat, difficulty swallowing, nausea, vomiting, sometimes including blood, diarrhea, and abdominal pain. The term dose is generally defined as the amount of a substance administered at one time. Yet there are several ways to describe dose in toxicology, including exposure dose, external dose, absorbed dose, internal dose, administered dose, biologically effective dose, total dose, and lethal doses 10, 50, and 90. Exposure dose is the amount of a substance encountered in the environment. External dose is a dose acquired by contact with contaminated environmental sources. Absorbed dose is the amount of a substance that entered the body through the skin, eyes, lungs, or digestive tract and taken up by organs or body tissues. Absorbed dose can also be called an internal dose. Administered dose is the quantity of a substance given to a subject. Biologically effective dose is a portion of an internal dose that is required to cause a health outcome. Total dose is the dose acquired by adding together all individual doses. Lethal doses 10, 50, and 90 is the dose that causes death in exposed animals at 10, 50, or 90%. Lethal dose 50, LD50, is used to compare the toxicities of chemicals. Lethality tests are rarely used in current research due to the availability of less destructive study methodologies. When studying the dose of a substance, toxicologists consider the total dose administered, the dosing time period or interval for multiple doses, and the body size of subjects. When multiple doses are administered over a period of time, the effect of the total dose may be different than when the total dose is given at one time. The toxicity of a substance is related to the body size of a subject. Therefore, children are often more susceptible to a dose that may be better tolerated by adults. A basic toxicology concept is the relationship between dose and response, or the correlation between the amount of a substance administered to a subject and the effects caused by that substance on the subject. Dose-response information may be used to demonstrate a causal relationship between a toxin and a biological effect, the minimum dosage needed to produce a biological effect, and the rate of accumulation of harmful toxin effects in subjects. Dose-response curves are graphs used to demonstrate the effects of exposure to a chemical or toxic substance on subjects. Dose-response curves can be shown for individuals or for populations. This is a simplified version of an individual dose-response curve for ethyl alcohol, starting with no effect, then with increasing doses, progressively developing symptoms of giddiness, sleep, deep sleep, unconsciousness, labored breathing, and death. The population dose-response curve is classically a sigmoid or S-shaped curve. This is also called a quantile curve. The response could be the percent of a population that has an observed outcome, like a protective or negative health effect, to various doses of a substance or chemical. At the beginning of the curve, there is a flat portion, demonstrating that at low levels, the dosage has no effect. This is the subthreshold dose. After the threshold dose has been passed, the curve begins to rise and then accelerates until it enters a linear phase where the response is proportional to the increase in dose. When maximal response is reached, the curve flattens out. As previously mentioned, A dose-response relationship is one of the indicators to assess a causal effect of suspected exposures on health outcomes. Environmental scientists have concerns over the long-term health effects of low-level subthreshold exposures to toxic substances. The toxicity of a substance is dependent on 5 factors: route of entry into the body, dose, duration of exposure, interaction with other substances, and individual sensitivity. We've already briefly discussed dose, but we'll now consider route of entry, duration of exposure, substance interactions, and individual sensitivity. This graph shows the relative speed of effect based on the route of exposure. The most rapid effect is through intravenous injection directly into the bloodstream, followed by inhalation via the lungs, intraperitoneal, subcutaneous, intramuscular, and intradermal injections, ingestion through the gastrointestinal tract, and dermal or absorption through the skin. The most frequent sites of exposure for environmental chemicals is by ingestion through the gastrointestinal tract, inhalation via the lungs, and absorption via the skin. The duration of exposure is important to toxicologists. Common terms used for exposure duration include acute, which usually refers to a single exposure for less than 24 hours, subacute for exposures for less than or equal to 1 month, subchronic for exposures of 1 to 3 months, and chronic for exposures of more than 3 months. Acute exposures may produce effects that occur but disappear rapidly. Workers, particularly agricultural workers, are often exposed to a variety of chemicals in greater concentrations for prolonged periods of time that can result in cumulative health effects. Most toxic exposures are associated with multiple chemicals, not just one. Factory workers are often exposed to a soup of chemicals. The 5 most dangerous industrial chemicals are chromium, zinc, ammonia, benzene, and mercury. When a worker is exposed to several chemicals, their combined effects may be additive, synergistic, antagonistic, correlative, or cause potentiation. Additive means the combination of chemicals produce an effect equal to the sum of their individual effects. Synergistic means the combined effect of multiple chemicals is greater than the sum of each chemical individually. One example of synergy is asbestos and smoking in causing lung cancer. Selikoff et al. Demonstrated that lung cancer mortality risk among asbestos insulation workers was much higher among those who smoked versus those who didn't smoke. Another example of synergy is the effects of solvents, N-hexane and methyl ethyl ketone. These chemicals together produce a greater impact on the nervous system than the sum of their individual effects. Antagonistic means that the combination of chemicals causes interference of the chemical's actions and effects. An example of antagonism is the negative or antagonistic impact of calcium supplements on the action of the antibiotic tetracycline. In a qualitative interaction, several substances that have no toxic effect interact to produce a toxic effect. Potentiation happens when one chemical that is not toxic causes other chemicals to become more toxic. An example of potentiation is isopropanol and carbon tetrachloride. Isopropanol by itself is not toxic to the liver, but enhances the liver toxicity of carbon tetrachloride when a person is exposed to these chemicals simultaneously. The responses of individuals to toxic substances can vary greatly. Factors associated with individual responses include age, sex, race, health status, genetics, medications, alcohol consumption, pregnancy status, underlying conditions, etc. For example, an individual with allergies may have a life-threatening anaphylactic reaction to the venom of a bee sting. Some individuals, when first exposed to a substance, become immunologically sensitized to it and can have a severe allergic reaction to a subsequent exposure. Adverse effects to chemicals include local effects, systemic effects, and target organ effects. Local effects relate to reactions where a chemical comes into contact with the body. For example, skin irritation. Systemic effects are defined as reactions occurring distant from the site of substance contact. Target organ effects are reactions confined to specific organs. The most common organs impacted by chemicals are the liver, lungs, heart, kidneys, brain and nervous system, and reproductive system. Latency refers to the time period between exposure and a measurable reaction and can range from a few seconds to decades. Injected substances like heroin can cause an effect within seconds to minutes. Most carcinogens, or substances that cause cancer, often have latencies of 10 to 40 years. For example, mesothelioma, a rare form of thoracic or abdominal cancer, has a latency of near 40 years from the time of exposure to asbestos or erionite, the main causal chemicals for this cancer. Long latencies between exposures and effects make studying these chemicals difficult. During a long latency of decades, the number of confounding variables, including exposures to other chemicals, can be astronomical and difficult to control for in a study design. There are three main methods for testing the toxicity of substances: volunteers who have had an exposure to a chemical, animal studies, and in vitro laboratory studies, including impacts on genetic material, often using bacteria, fruit flies, animal or human cell line cultures, etc. In the United States, the Food and Drug Administration is responsible for determining the safety of foods and new medications. There are 5 steps to the approval of new drugs in the United States. 1. Identification of a potential new drug. 2. Preclinical research on animals to ensure safety. 3. Clinical human trials, the first 3 phases of clinical trials. 4. FDA review and approval for use in humans. 5. Post-market safety monitoring, 4th phase of clinical trials. Let's briefly review the 4 clinical trial phases. Phase 1 clinical trials focus on the study agent in a relatively small number of healthy subjects, approximately 20 to 100 in number. The goals of Phase 1 trials include looking for safety issues, including side effects, and evaluating how the agent should be taken. In vaccines, this includes the gathering of dose-related immune response data. Phase 2 trials also evaluate the study agent for safety, but in a larger study group, usually a few hundred up to 1,000 in number. For a medication study, patients are those with the condition for which the medicine, device, product, or treatment has been developed. For vaccines, the study group is generally healthy individuals. Researchers in Phase 2 trials assess the short-term safety and effectiveness of the drug or vaccine, explore drug or vaccine interactions, and find the dose at which the agent works best with the least side effects, and conduct a small-scale placebo comparison. Phase 3 trials look at a larger group of individuals, often 1,000 to 20,000 individuals that better represent the health of the population. Phase 3 trials aim at confirming safety of the agent, evaluate efficacy in this larger population group, compare the new agent to other compounds, for example, versus a placebo or other therapies, define risk benefits, and gather information for a licensure application and package insert. Following the completion of phase 3, a drug or vaccine is approved or not approved by the U.S. Food and Drug Administration, FDA, for use by specific patients. Phase 4 may only begin if the drug is approved. 12% of medicines that are evaluated in clinical trials will be licensed. Finally, after the drug is approved, researchers look to study the effectiveness of the drug in a wide variety of patients, as well as monitor the safety in a large group and enable the development of new uses for the compound. These studies often involve thousands of individuals from the population using the licensed drug or vaccine. Phase 4 studies often inform future research and development. Phase 1 to 3 trials are often not of sufficient numbers to appropriately evaluate intermediate or rare adverse events. Due to the large number of study subjects, Phase 4 studies can expand the safety profile of the drug or vaccine by evaluating that larger study group for intermediate or rare adverse events, along with safety in subpopulations. This phase of studies also address any emerging safety issues that arise with drug or vaccine usage. As mentioned previously, Phase 4 studies relate to post-licensure safety monitoring, where Phase 1 to 3 studies address the pre-licensure and licensure safety monitoring process. Before the rigorous use of these clinical trial phases, drugs were being marketed to the public without adequate evaluation of safety. For example, in the 1950s, a drug called Thalidomide was being used in Europe, Japan, Australia, and Canada to treat morning sickness in pregnant women. This drug was not approved in the United States due to lack of safety studies. As a result, more than 10,000 women treated with thalidomide gave birth to children with severe birth defects, including missing limbs, also called phocomelia. Once toxicology information for a substance is available, a health risk assessment helps determine an appropriate response regarding that substance. Risk assessment generally is associated with 4 assessment steps. 1. Hazard. 2. Dose response. 3. Exposure. And 4. Risk characterization. A hazard is defined as the ability of an exposure to have an adverse effect and may result from chemicals, biological agents, physical and mechanical forces, and psychosocial impacts. Dose response, as previously discussed, is the correlation between the amount of an exposure to a substance and its effects. Exposure identifies the population exposed, the demographic composition, size, the magnitude, frequency, and duration of the exposure. Risk characterization develops estimates of the number of excess health events expected at different time intervals at each level of exposure by summarizing the information from the hazard, dose response, and exposure assessments. The meaning of health risk varies between individuals and groups of individuals. For example, health professionals versus laypersons, etc. Health professionals often characterize risk in terms of mortality. Laypersons often characterize a health risk based on perception scales of hazard concern and knowledge concern. A low hazard perception score is associated with a toxic exposure, for example, being controllable, not global, no or low mortality, low risk to future generations, and the risk declining, versus a high hazard score associated with not being controlled, global in scope, high mortality, high risk for future generations, and risk increasing. A low knowledge score is associated with a toxic exposure that's observable, known by those exposed, and has an immediate effect, and known to science, versus a high score associated with an exposure that's non-observable, unknown to those exposed, delayed effect, new risk, and unknown to science. Toxicology risk assessments provide information for public health and policy decision makers. In summary, a basic assumption of toxicology is that all substances are poisons. Environmental toxicology is a multidisciplinary field of science concerned with the study of the harmful effects of various chemical, biological, and physical agents on living organisms. A dose-response relationship is one of the indicators to assess a causal effect of suspected exposures on health outcomes. Adverse effects to chemicals include local effects, systemic effects, and target organ effects. Latency refers to the time period between exposure and a measurable reaction and can range from a few seconds to decades. In the United States, the Food and Drug Administration is responsible for determining the safety of foods and new medications. Phase 4 clinical studies relate to post-licensure safety monitoring, where phases 1 to 3 studies address the pre-licensure and licensure safety monitoring process. Once toxicology information is available, a health risk assessment helps determine an appropriate response. The meaning of a health risk varies between individuals and groups of individuals. Toxicology risk assessments provide information for public health and policy decision makers.